talk a little bit about his research at MUSC.
All right, so thank you all for coming to my presentation on the protease activated receptor-1 and its mediation of calcium ions within the mesangial cells. So to first understand my research, we have to know what the kidney does within our bodies. So the kidney's primary functions are maintaining mineral levels within the body. It helps filter the blood, maintains glucose levels within the body, and it can also create urine too. Now, the kidney is made up of nephrons, and within these nephrons are the glomeruli, or glomerulus for singular. So the glomerulus is made up of multiple kinds of cells, being the periodal epithelial cell, the podocyte, the mesangial cell, and then the endothelial cell. So essentially, the glomerulus is just a vein that creates a loop with all these different cells surrounding it, with the periodal epithelial cell surrounding the outside, creating the Bowman's capsule, creating the outermost structure of the glomerulus. The endothelial cell being the actual thing that the veins are made of, and the podocytes being what controls the filtration. So for today, we're going to be focusing majorly on the mesangial cell. So the mesangial cell is muscle-like in nature, to where it expands and contracts to help blood pressure. And it can also create an actin mesh to work almost as like a fish net to help further filtrate the blood within our bodies. Now, the mesangial cell has many glomerular pathologies associated with it, including diabetes and chronic kidney disease, which is going to have a heavy focus in this research. So on the mesangial cell are the protease activated receptors. What we're going to be looking at is protease activated receptor 1, or PAR1. This receptor's agonist is protease, and the most common protease here is thrombin. It's a G protein coupling receptor, and also when stimulated causes a calcium ion influx. This is a relatively new discovery, the PAR receptors, being discovered in 2000, with its structure only being discovered in 2012. So it's relatively new, not much is known about it. So the purpose of this research is to figure out the relationship between the PAR1 receptor and chronic kidney disease, which is a disease that's incurable and will eventually be fatal. So how we're going to figure out this relationship between the two are through two Nobel Prize winning methods, electrophysiology and confocal imaging. Electrophysiology is essentially creating a micropipette small enough to where you can latch onto one singular ion channel and monitor its ionic activity. For us, it's going to be the calcium ion. And for confocal imaging, what we do is we create two groups, the control group and the high glucose treatment group, which is a diabetic situation. And then what happens is, especially for the diabetic situation, the high glucose treatment, what we do is we incubate that for 12 hours. So it has essentially diabetes, or at least will simulate diabetes. And after that, we add the dye, fluorescent A, and we let that soak into the cells for about an hour. Once the dye is soaked in, we change the solution to make sure that no leftover dye affects our imaging. And then we image our cells to see what kind of results we have, what kind of calcium influx is happening. So to begin with, we're going to look at a control group for confocal imaging. The drug that we're using to activate the cell is TFLR. And what happens is we can see the baseline of basically very little calcium response happening, where then we add TFLR, and there's a high influx of the calcium ion, which then slowly dies down and becomes about 0 again. So this is a healthy, non-diabetic cell, and this will not lead to chronic kidney disease. But here we have a high glucose treatment group, or a diabetic situation. And as we can see, there's going to be a high response to the TFLR, which does not die down quickly. So since this has an extended calcium influx that's not going down, what's this going to lead to is it's going to lead to cell apoptosis, which essentially leads to chronic kidney disease. here is the electrophysiology, which is essentially a different method of proving the same thing. So we can see our baseline of what the cell is, but then once we apply TFLLR, then there is an extended calcium response that never truly dies down and goes back to baseline. This is just explained by the drug takes a little bit of time to reach the cell, but once it reaches the cell, there's the extended calcium influx. So this calcium influx, like I said before, leads to cellular apoptosis or even remodeling of the cell, which also will lead to chronic kidney disease. And this is only in the diabetic situation groups. The control groups are completely fine. And that's the microscope that we use for electrophysiology. So in conclusion, what we figured out is we now know the functional presence of PAR1, which is to release calcium within the cell. We also know that when we activate PAR1, that it creates more PAR1 receptors, which amplifies further we go along. Then we know that the calcium influx will lead to the cell death. And finally, we now have a connection between the PAR1 receptor and chronic kidney disease. Places we can take this research further is we can do further studies on the other renal cells, the glomerular cells. So say podocytes, we could do the same experiments on them. The periodal epithelial cells, the same experiments on them, and see what results come out with them. We can also find a blocker for the PAR1 receptor, so we can try and make a cure for chronic kidney disease without just replacing the kidney or having the fatality. I'd like to thank MUFC and GSSM, but especially Dr. Oleg and the Paligan lab. Plus, I'd like to thank Dr. Riddle, thank you. And any questions?